Our next talk will be, again, a partner presentation, professor and grad student. For those of you who were here last year, you may remember a short presentation from Basim Abbasi, who's a professor in engineering, who'd been working and gave some preliminary results on research they had done at looking at various ways of processing liquid whey, leftover, not to say waste, from cheese making. And after looking at some higher tech solutions, they got to a lower tech one that looked really promising. And this is a follow-on, so you'll hear a little teaser from that. But what happened next? Pregnant pause for drama here. But looking at this actually feeding back in a circular way into feeding cows. So it's a pleasure to introduce Tatum Schooley and Katie Wood from Department of Animal Biosciences, who will tell us a bit about not 4 R's, but 3 R's this time, on using milk processing byproduct as a feed for cows.
Good afternoon, everyone. As Stephen mentioned, this has been a kind of interesting journey looking at reusing some of these byproducts. It all started about 5 years ago, where we were in the middle of the pandemic, and we were approached by some folks at OMAFA trying to solve some of the issues that some of the small artisan cheese factories in Eastern Ontario were having, where they were shipping their waste whey at over $1,000 a truckload, with no alternative ways to get rid of it in Eastern Canada. They were approached by some feedlots to look at some feed value, which is why the beef researcher is here and how I got involved. And then from there, we did some studies, and it was in the middle of the pandemic out in the manure storage shed because we couldn't get within 6 feet of each other. And we're looking at some of the nutritional value in it and found that although there was lots of good nutrient in these byproducts, the water level was quite high, which means if we were replacing some of the commodities, it would be pretty soupy. So with that, we ended up being paired up with some folks in engineering that were looking at wastewater treatment from some of these plants. And so we kind of joined up together in looking at trying to dewater some of these milk processing byproducts, which is the presentation you heard from Basim last year. And this is kind of evolved into a multidisciplinary research group, including Giselle Lapointe and Alice Marciniak from Food Science, Ping and Basim, and Ryan in the Department or College of Engineering, I should say, and then ourselves on the food side or animal feed side, looking at some outcomes for some of these projects. And as Stephen mentioned earlier, it resulted in a really nice award a couple weeks ago for Dairy Farmers of Ontario, bringing these groups together and solving a big industry issue. But today I'm going to turn it over to my master's student Tatum, who really knows more about this project than anyone, and tell us a little bit about some things that we've learned in looking at waste milk byproducts as a feed ingredient for cattle. So I'll turn it over to you, Tatum.
Thank you, Katie. Well, hello everyone. My name is Tatum Schooley, and this is my master's thesis, Reduce, Reuse, Recycle Milk Processing Byproducts as an Alternative for Corn in Dairy Cow Diets. Canadian milk is used to make several products, including cheese and butter. However, this process results in a large volume of byproducts, including solids non-fat, or SNF, and cheese whey. About half of these products are used in other food or feed production, and some are used across other industries as well, but disposal of the surplus presents some environmental concerns. SNF and whey make valuable feed ingredients, but the low dry matter complicates handling, transport, and limits applications. Other current methods of dewatering, while effective, can be cost, time, and energy consuming, which can limit the large-scale application possibilities. We've been investigating bentonite and nanofiltration to replace the ultrafiltration pre-filtration steps before nanofiltration dewatering. So we've been working with the engineers from our multidisciplinary committee to develop this protocol to kind of separate the different components of SNF a little more cost effectively. And we found that using bentonite adsorption to replace ultrafiltration before nanofiltration dewatering produces a whey protein plus bentonite slurry, or WPB, and a separated lactose supernatant, or SEPLAC. So the bentonite clay adsorbs or kind of adheres to the whey proteins, and that complex settles to the bottom of the solution, which can then be separated from the larger lactose supernatant there. That supernatant is then run through nanofiltration, which concentrates the solution and removes filtered water, leaving a concentrate of about 15% dry matter. Now, these byproducts, these separated byproducts, could actually have some health benefits in addition to providing an outlet for SNF surplus, as lactose, the major component of the supernatant, has been shown to increase maintain or increase rumen pH, increase butyrate, and decrease propionate concentrations compared to high-starch diets in dairy cows. And bentonite, which we are using to bind and separate out the milk proteins, has been shown to buffer pH against sudden changes due to fermentation in the rumen. This lactose could help prevent subacute ruminal acidosis or diet-induced milk fat depression, which could result from reduced intake and pH caused by excess starch fermentation going on during those high-starch diets.
So now we've kind of got a product that, at least on paper, nutritionally looks like perhaps high moisture corn. But there's still a lot of technical things we had to figure out to how to get this into a dairy type TMR. So Tatum did a lot of work, kind of sorting some of these out before we could proceed with our animal trial. So she's going to tell you about some of those.
All right. So for our preliminary experiments, we hypothesized that whey dewatered with bentonite and nanofiltration could replace high moisture corn up to 12% in a TMR without experiencing any significant liquid seepage from the diet over a 24-hour period. Our objectives were to determine the maximum inclusion of the liquid ingredients in the TMR, to estimate the nutritive value of lactose and that whey protein bentonite relative to high moisture corn, and to assess pathogenic microbial growth from whey byproducts. This protocol was developed with the engineers, but we developed a method for using cheese whey sourced from Guns Hill Artisan Cheese, and then mixing in 30 grams per liter of a sodium bentonite clay using a mixer attached to the top of a tote. This mixture was allowed to settle and separate over a period of about 4 hours before being separated with a pump, and the ingredients were kept frozen until use in the trial. You can see during the separation, we had 2 distinct layers formed of the separated lactose supernatant and then the whey protein bentonite slurry on the bottom, both after the initial separation and then after thawing. These are the results of our nutrient analysis of our whey protein bentonite, separated lactose, and the synthetic lactose we used in our experiments. We found that they all had similar available energy to high moisture corn, which you can see in the bottom row here with the total digestible nutrients, or TDN, about similar to corn. Since we had to do our whey separation at room temperature, we also did a microbial analysis of our different products at various stages during that process, just to make sure that everything that we were feeding to the cows was safe and clean and didn't contain anything bad in it. But we tested at various points during the separation and storage and afterwards, and found no coliform, E. coli, listeria, or salmonella at any points during our process. So our separated ingredients were completely safe to use. For our liquid seepage experiment, we mixed the regular lactating cow TMR used at the dairy barn with 12 to 16% synthetic lactose, or Synlac, replacing high moisture corn. This synthetic lactose was mixed to a concentration of about 15% to simulate the dry matter of the nanofiltration concentrate. We mixed the diets and then stored them in buckets with 129 holes drilled into the bottom to resemble the bottom sieve of a Penn State particle separator to allow any liquid not retained by the ration to seep out the bottom, be collected and measured after that 24-hour period. So after those diets were mixed, we stored them in these buckets and left them there for a period to confirm that all of the nutrients we added with that liquid lactose were staying inside the ration. And then we confirmed our results in the large-scale mixer and feed bunks as well. From this liquid seepage experiment, we found that the liquid lactose ingredient can be included up to 15% in a TMR diet without experiencing any significant liquid seepage over a 24-hour period. So overall, our preliminary results found that separated lactose, synthetic lactose, and WPB had similar available energy to high moisture corn. We found no pathogenic microbial growth in any of our samples. Synthetic lactose can be included up to 15% in a TMR without experiencing any significant liquid seepage. And we experienced no liquid seepage in the mixer or the feed bunks when scaled up to 150 kilograms.
All right, so now we have a product that at least looks like corn on paper, and now we need to evaluate what that actually does within the animal. And so we did some test mixes at Alora. We were concerned that it was going to be really sticky as well in the TMR mixer and in the bunks. We found that that wasn't the case, and so we were going to proceed with our animal experiment, which just finished less than 2 months ago. And so we've got some preliminary results to share with you for now, and many more results yet to come. So back over to you, Tina.
All right, so for our animal trial, we hypothesized that since the nutrient composition between these products was similar, high moisture corn could be replaced up to 12% with lactose and that whey protein bentonite without impacting milk production, composition, or rumen health. Our objectives were to observe the effects on milk production and composition, serum metabolites, rumen pH, and VFA concentrations, and to estimate the nutritive value of lactose and WPB relative to high moisture corn. For this experiment, it took place at the Alora Dairy Research Station between May and September of this year. We used 10 multi-paris mid-lactation Holstein cows between about 80 and 100 days in milk. And they were organized in a replicated 5x5 Latin square, blocked by early and late days in milk for this experiment. We did 5 21-day periods, each of which included an adaptation to the diet and sampling of all of our different parameters. For each period, so we had about 2 weeks here where the cows were adapting to the diet. And then in that last week, we measured or collected a number of things, including milk yield and milk samples, feed intake and diet sorting, body weight, body condition score, rumen fluid, fecal, and blood samples. The treatment diets included 0% or a control, 4, 8, and 12% synthetic lactose, and an 8% synthetic lactose plus 2% whey protein bentonite diet, all replacing high moisture corn. Same deal here, we use the synthetic lactose mixed to 150 grams per liter to simulate the concentrate dry matter after nanofiltration, and our control diet was just the base lactating diet used at the barn. So you can see here our different treatment diets. The HMC, high moisture corn, about halfway down is decreasing through our treatment diets, and then you can see the synthetic lactose and the whey protein bentonite at the bottom increasing through our treatments. So to mix these diets, the whey protein bentonite and high moisture corn were weighed out daily, and the synthetic lactose was mixed daily, stored in a drum, and delivered into the mixer via pump and hose. We took feed samples weekly of each of our diets and each of our diet ingredients, and then the last 4 days of each period, we measured fresh diets and orts to measure nutrient composition as well as sorting via the Penn State particle separator. We took fecal samples in the last 3 days as a measure of digestibility, and for milk yield was recorded by the rotary each day, and samples were collected AM and PM towards the end of the period. Body weight and body condition score were measured between periods as well. Blood samples were collected from the tail and used to measure glucose, urea, NEFA, and BHB. And rumen fluid samples were collected measured for pH, and then frozen in liquid nitrogen for later VFA analysis. For our results, we used contrasts in SAS to look at the differences between the different inclusions of lactose and then the treatments with or without bentonite. So you can see here kind of for our results. On the left here, we've got our lactose graphs where you can see the 4 different inclusion levels of lactose, 0 to 12. And then our p-values for our linear contrast and our quadratic contrasts on that graph there. And then on the right side there is our bentonite inclusion graph, is looking at that 8% lactose diet, either with or without the whey protein bentonite. And the orthogonal contrast p-value is in the middle of that graph there. So as you can see from this slide, we had no significant impact of bentonite or lactose on body weight on any of our cows during this trial. We saw no significant differences in body condition score either. Milk yield was unaffected by either of these treatments, as well as milk fat was unaffected as well. We did see a significant linear increase in milk protein with increasing lactose in the diet. And we also saw a significant linear decrease in milk lactose with increasing lactose in the diet. We found that energy-corrected milk, calculated using the Shanja 1990 equation so that we could include lactose in these calculations, also saw no significant differences between our treatment groups. And rumen pH, we saw no significant differences either, though all of our rumen pHs during this trial maintained a healthy, normal range during our spot tests. So as we just finished our trial a couple of months ago, we still have lots of results and sample processing ongoing. But we've got several results to come, including things like intake, sorting, diet analysis, digestibility, serum metabolites, and rumen VFAs. Though I can say, hot off the press, we did recently get some of our serum results back, and it looks that urea as well had a significant linear decrease with increasing lactose in the diet, which kind of coincides with our milk protein results, and is very exciting to see what else comes out, though we can't really make many conclusions yet without the rest of the context of our remaining data. So in summary, we found that liquid seepage occurred over 15% inclusion of that liquid lactose ingredient in a dairy cow TMR. We found a linear increase in milk protein and a decrease in milk lactose with increasing lactose in the diet, while milk yield and milk fat were unaffected. We found that cows maintained body weight, body condition score, and rumen pH during this trial, and we've seen no significant effects of bentonite so far. So recycling these milk processing byproducts into feed could find a method to reduce food waste from the dairy industry, as well as provide a sustainable method for disposal of SNF and whey surplus. Finding cost, time, and energy efficient techniques is going to be key to facilitating these methods widespread and for large-scale applications. This method could provide producers with feed ingredients that are locally sourced and dewatered to minimize transportation costs, could provide an alternative feed to traditional grains when they become more expensive, could improve rumen health compared to high-starch diets, are more widely applicable across various industries with a higher dry matter, and promote a circular economy. So in short, so far, we found that dewatered milk byproducts can be included up to 15% in a TMR without experiencing any significant liquid seepage. And we found that lactose inclusion in a dairy cow TMR was found to increase milk protein and decrease milk lactose while maintaining milk yield and rumen pH. I would like to thank DFO, OMAFA for funding this project. I'd like to thank all of our collaborators in our multidisciplinary committee for helping us develop this project. I'd like to thank all of the staff at the research stations for helping me out during this trial, as well as Katie and all of the rest of our people in the rest of the people in our lab for helping us all the way through. And thank you to all of you for listening.